Okay, we're going live now.
Live on YouTube. It's not like Saturday Night Live, but it's different live. Good.
Good. Let me share my screen. So,
Can
you see it?
Yes, yes.
You're still in the practice section, just so you know.
Oh, okay. No, yes, you're right. We haven't started the webinar. We only have the link. Oh, that's good. I thought that once you do one, you can. But anyway, this is all online now, so it's as good as live. It's only that. Yeah. Yes. Might as well just let people in the room anyway. So we just wait a minute or two to make sure that people have enough time to join the room and be prepared for your talk and see your beautiful introduction slide with what I imagine is something you draw.
Yes, I drew it by myself in iPad.
Yeah, I thought this looks like iPad art.
Yes, it's iPad. My computer was broken down 1-2 days and I need to give a talk. So that time I drew this.
So you're actually giving this talk through your iPad? No, this is your Mac. This is your Mac.
That was before, yeah.
Oh, we even have Arturo together today.
Slides, or you also see yourself in my screen?
No, I only see the slide.
Okay.
I also see your doc, so I see your icons. If you get Slack messages, we might see those as well. So you might want to hide that.
Yeah, I just quit. Yeah.
Yeah, there's always a way in Macs to go to this sort of option in the top right corner of the screen where you can sort of deactivate notifications, so you don't get notifications in the middle of your.
I see.
So we have a few people, so maybe it's a good time to start your talk, Weini. Let me just give a brief introduction, and then you'll be good to go.
Okay, good.
So, ladies and gents, it's my pleasure to be introducing today Dr. Weini Huang, who is joining us. Although the slide says Tampa, is actually currently in London, where he is a senior lecturer in mathematics in the Institute of Mathematical Sciences at Queen Mary University of London. As a lecturer, senior lecturer now, right?
Yeah.
Yes. So obviously, for those of you who are not familiar with this is, I guess, similar to associate professor here in the United States. Yes. Before she joined Queen Mary, Weini did her PhD at the Max Planck Institute for Evolutionary Biology under Anne Traulsen in beautiful Plön. I guess after that, you moved to London, where you work at the Barts Cancer Institute in London as well. And since then, I mean, even at Kiel, you were actually working on evolutionary mathematical models. And now you're also working, since you moved to London, on applying these evolutionary mathematical models to understand cancer evolution. So you talk today is in mathematical models of evolutionary medicine, and I think that there's going to be genetics and chromosomal segregation today. So I'm definitely looking forward to your talk, Weini.
Okay, thank you, David. Thanks for the invitation. So I'm very happy to present actually our still going on project. I mean, we kind of finished in the submission process. So the paper manuscript already on bioRxiv if you are interested. So
archive.
So we are talking about ECDNA. So before I go, I want to thank our team. The major person in the team is the theory actually started with Yuri and Benjamin. So we had a theoretical project actually from a seminar like this. And we're talking about how to work. Yuri actually has the background of evolution of multicellularities, like how cells evolve from single cell to multi-cell organisms. And then we are talking about how this will relate to some work we have seen in cancer evolution. And this link up to our theoretical project. And then later, when we had our theoretical project already out from the back of 2019, at the end of 2019, and we actually later meet all the experimental people in the conference, and we kind of teamed up together now. So I think from the audience background, everyone kind of agreed that cancer evolution actually is a clonal evolution that relate to that cells are reproduced. So the information carried from the mother cell to daughter cells. So there are lots of common things about cancer evolution related to classical somatic evolution in biology. Actually, ECDNA is as part of the information in the cells also follow this somatic evolution. So what is ECDNA? So ECDNA actually often arise from chromosomal instability. So when the cell reproduce, some parts of the genome can't just break to pieces, and then they're formulating a circular DNA structure in the cells. So this leads to the original of the ECDNA. So if the difference of the ECDNA with chromosomal DNA is that in the chromosomal DNA, we have the centromere. So when the cell divides, the information was kind of equally partitioned between the two daughter cells. But ECDNA don't have this centromere. So instead, they have this random segregation, so which lead to a kind of diversity of ECDNAs among cells. If you look at my picture here, can you see my pointer? So if you look if you look at these cells, the original cell might have only one ECDNA copy, and then there are different partitions that are happening in the process. So it can be that both daughter cells inherit one copy, but it also can be that one cell inherit no copies, but the other daughter cell inherit both copies. And then with further, when the cell has two copies, then you have more possibilities of partitions in between the daughter cells. So this leads to a diversity of ECDN copies among the cells. And as the cells, then you can divide the cells to that those cells carry the ECDNA, which is in the red box, or those cells don't carry the ECDNA. And as usual, it follows some evolutionary process with somatic evolution. For example, if you have number one cells arranging with these ECDNA copies, but also the daughter cell can lose it. So you have some linkages that have ECDNA diversity and some don't have it. So what is interesting is that ECDNA actually originally in old times, that back in the 50s already people talking about double minutes, which is two ECDNA rings structures. So people see it in the staining, but they don't really call this as ECDNA. But as ECDNA become a new hot topic in cancer evolution, and actually people studied, look at the TCGA data and look at all these cancer types and found out ECDNA actually is not rare. So if you look at this picture here, this is published actually last year in Nature Genetics. They found out that many cancer types have ECDNA, which is represented by the red bars, especially in glioblastoma. More than 50% of patient samples has ECDNA, followed by esophagus, ovarian cancer. So in some other cancer types, you might have few. For example, in colon cancer, it has less ECDNA, less samples at ECDNA, but still is more than 10% of the samples. So even more, what they notice is that, so they have developed some bioinformatics tools to recognize the ECDNA and also the chromosome DNA, and found out that those samples carry the ECDNA, which is marked with red again here, with circular. They actually has worse survival probabilities for the patients. So how does ECDNA drive the tumor evolution? Mainly through the oncogene amplification. So in the same paper, they look at in which genes kind of carry this ECDNA. They found out, for example, MDM2, which is kind of related to TP53, and you have MUC gene and the EGFR genes that are most pronounced, dominated in the data with these oncogenes that being amplified. Moreover, if you look at the right side panels, which is in our bioarchive paper, and they look at these cell lines. So SNU16 is one type of stomach cancer, and they found out that the distribution of these ECDNA copies, that some of them can reach, each single cell can have highest as 100 or 150 copies. And in the patient data, is also the highest number of ECDN copies can reach 30 or 50. So you can see that if this is driven, the oncogenes, so it's very intuitive to say that the cells carried ECDNA will amplify oncogenes. And because they have these ECDNA copies, so they don't only have 2 copies, but they can have multiple copies. And this will bring a fitness advantage, which might lead to cancer. But of course, this is relatively hard to measure directly in the lab and most likely impossible in the patient. So our questions, how can we bring together with our theoretical work with the data to test and quantify whether those ECDNA cells are really positive selected? So to do this, so we introduce stochastic model to model the ECDNA dynamics. The reason is because this random segregation process in ECDNA that I was talking about. So in this process that we have, for example, if we have one cell with 2 ECDNA copies, during the cell doubling, cell division, the ECDNA information will be doubled. So there will be 4 copies, and each copy has a likelihood to go one of the daughter cell with probability P. So if this P value is equal to 0.5, then the distribution of the ECDNA copies among the 2 daughter cells will follow a binomial distribution around the peak. So for example, if the mother cell has 10 copies, then the distribution of the 2 daughter cells will be almost like a Gaussian distribution around 10. So when we have this random segregation, when we look at back to the data, we can use this fish staining to check it. So the theoretical prediction is that if the p-value, so the likelihood each single copy go to one of the daughter cell is 0.5, then it's Gaussian distribution around 0.5 for the fraction of the copy numbers between the 2 daughter cells. Anything deviating from 0.5 will lead to a distribution with 2 peaks. So now we work with the experimental people, mainly with Paul Michel's lab. So they have different cell lines. So the SNU is the stomach cancer. Then you have the GBM is the glioblastoma cancer, cell lines, and PC is the prostate cancer, and the colon is the colon cancer. So they try in different cell lines, and they want to stain the ECDNA copies. So you can see the red colors are the ECDNA signature, and the green ones is the centromere. So what they did is that they have 200 pair of daughter cells, so 100 cells, let them divide, and then record how many ECDNA copies between the 2 daughter cells. And actually, from different cell lines, they more or less confirm that this value, so the P should be around 0.5, because you can see that there's kind of a peak around 0.5. So we get encouraged that this is such a relatively simple stochastic process, and we want to bring this assumption of random segregation to see what is the properties that we actually can measure in the data that we can theoretically predict or simulate it. So first thing we look at is the fraction of cells with or without ECDNA. As I said, in this somatic evolution of ECDNA, the cells divide, so the daughter cells can carry equal partition, but you can also have cells loose. So which means there's always a fraction of cells that don't carry ECDNA during the random segregation. So we group these cells don't carry ECDNA as Fn-1, which is just n-1 divided by the total number of cells, and the rest of cells as Fn+1. So we have a stochastic process, but first we want to see whether we can analyze some solutions through the deterministic dynamics. So of course, with this, we need to assume what is the population dynamics here. So we look at the branching process, which means that we have a growing population. The reason is that because from the cell line data, also from the patient data, most ECDNAs are found in growing in tumor cells. So if it's normal cell lines, or they don't see so much of ECDNA. We can discuss why this led. So for the branching process, so then we are modeling a growing population, and we have another assumption is that the fitness of the cells carry ECDNA is copy number independent. So which means we assign a constant fitness for the cells carry ECDNA. No matter it carries 10 copies or 1 copies, they have the same fitness. So this is just the first attempt with the simplest assumption. So it's no reason that we have to assume in this way. And that's some further work we need to do as well. So to do this, then we can start with our basic deterministic dynamics. So here, n minus t is telling is the number of the cells not carry any ECDNA changing over time, and n plus t is the number of cells carry ECDNA. So we group the cells with these two categories. And of course, the change this over time is the dynamics itself, and plus v times m plus t. So the v here actually is an unknown function that we assume that is the loss rate of the ECDNA due to complete random segregation. So for example, if we have a cell with 2 copies, then with this VNT, n plus t, it just tells you that how likely these 2 copies get totally lost in one of the daughter cells. So we don't know this function, but what we want to do is to get rid of this v that we use in these differential equations. So then f is fn minus 1, which is the fraction of the cells not carry ECDNA. And we can write down this equation just by rearranging the equations in the box, that we found out that the derivation of the f over t is just equals to 1 minus f times the Vm. So when we rearrange these equations, we kind of can, write down the solution for n plus t and n minus t by plugging one of these computational simulation results we have here. So up to here is all analytical, but we have this n minus t and n plus t equals 1 divided 2 to the t come from our stochastic simulations. So we don't have a solution for here, but we have computational simulations we plug in, then we can get our neutral solution. So the neutral solution shows that the fraction of the cells carry ECDNA is increasing with, if you look at the, let me just show the figures. So these 2 figures shows the number of cells carry ECDNA and the number of cells without ECDNA. So the gray one is the number of cells without ECDNA, and x-axis is number of cells. So because we are looking at a branching process, so the number of cells kind of indicate in time. So when the time increases, the number of cells increases. So you can see that the absolute value of number of cells with or without ECDNA, of course, both increases because that we are looking at a branching process, but the fraction is different. So the solution under the neutral selection, what we see is that the cells carried ECDNA is 2 divided by 2 plus n to the n. So which means under neutral selection, the cells will slowly lose the ECDNA the fractions. So more and more cells will not carry ECDNA because the cells not carry ECDNA is kind of absorbing state of the system. Once you lose the ECDNA, you don't get it in the process because it's a rare event. So if we look at the same system, but we introduce selection, which is S here, so we can write the same differential equations. Instead, we have also S. So S equal to 1 is neutral selection. S equal to 2 is positive selection. So everything that larger than 1 is positive selection. So this, but however, we can't really write down the exact analytical solution for when we have introduced the selection. So instead, we have numerical simulations to simulate our deterministic system, and we have the stochastic simulations to individual-based simulation to confirm it. So in this case here, we can see when selection is neutral, which is S equal to 1, so the fraction of cells without ECDNA is increasing over time, as what I said before. So the absorbing state is the cells without ECDNA. But if selection is positive, then we can see a kind of first increase, then decrease. So the fraction of the cells without ECDNA is increased first and decrease again. So in the end, you're ending up with more cells carry ECDNA if it's positive selection. So this is confirmed both by the numerical solutions from our differential equations in the deterministic system, also by the stochastic simulations with individual-based model, where we only have the assumption of random segregation and constant fitness. So if we look at our cell line data, so there we have looked at different cell line data. So we have looked at glioblastoma data, we have looked at the neuroblastoma data, which is kind of cell line derived from patient data. And then we have different cell lines with different oncogenes in the ECDNA. And we found out actually all these cell lines show a positive selection signature. So what you can see is here, if you look at different cell lines, the dashed line here is the stochastic simulation for fraction of cells with ECDNA, which is very low if you have S equal to 1, but if you have S larger than 1, then the fraction is much higher. And then you can see all the data shows much far away from the theoretical prediction for S equal to 1, which means the cell line can have a higher fraction of cells with ECDNA because the selection is positive. So this is when we look at like the fraction of the cells with ECDNA copies, but we can look at more details. So we can look at not only grouping the cells with N0 and everything else with N+, but we can look at cells carry 0 ECDNA copy, which is N0, cells carry 1 copy, 2 copies, 3 copy, and so on. So we use NKT denote the number of cells carry K ECDNA copies. Then we can write down the stochastic, the master equation of the system. So you can see that again, the change of the NK is related to itself, because once the cell divides, so you lose to be exactly have K copies, but the daughter cells can have a certain range. So if a daughter cell want to have at least a K copy of ECDNAs, of course, the mother cell should at least have K divided by 2. So because if the cells is ECDNA copies are doubled and all come to the same daughter cell, the maximum you can get K is that you have minimal K divided by 2 copies in the mother cell. So in this part, you can see the binomial distribution play a role in parts of this equation here in considering how much the daughter cells get how many copies. So if we write down the same equation in a continuous limit, then we can counting with integrating all the daughter cells starting from K divided by 2 to infinity, and how do they contribute to the daughter cell. So this master equation, we can't really analytically solve it, but we have some approximations that can be useful and also can confirm with our computational simulations. So I will go through by some points that we think is interesting to distinguish positive and neutral selection, or some properties maybe the same for positive or neutral selection. So the first conclusion we see from this stochastic process is that the distribution will be wide and the tail follow a combined exponential and the power law decay for any selection intensity in our model. So again, the x-axis is the ECDNA copy number and the y-axis is the frequency. So how many in the whole cell population, how many cells carry a certain copy number? And we found out the tail is very wide. And if we look at our cell line data from our collaborators, so again, they have different cell lines, have the colon, prostate, glioblastoma, and the stomach cancer. And we found out all of the cell lines actually has a really wide distribution. And if we run our simulations with different parameters for different cell lines, we kind of can find a really good fit from the computation simulation with the distribution. So the second thing that we see is measuring the mean and the variance of the distribution. In our cell line experiments, for example, we can use the fish data to look at, count the copy numbers in each single cells. But for example, for example, in patient data, then in some of these cases, you might not able to see the single cell copy numbers. So it's very useful to look at some mean values, how do they change? So in the mean ECDNA copy numbers, that we see that if the selection is neutral, which is S equal to 1, then the mean copy number is a constant value. So if we started with one cell with one copy number, then the mean number will stays at 1. But if you start with a population on average has higher ECDNA copies, then stays in that constant as well. But the mean ECDNA copy number, if selection is positive, then we can see that it increases actually with time. And again, if we look at the variance of the ECDNA copy number, we notice that both variance will increase over time, whether selection is neutral or not. But if selection is positive, then the variance increase in a much faster compared with the neutral case. Another thing we see is using the Shannon diversity to measure the ECDNA copy numbers in the population. So the Shannon diversity actually is quite simple. If we have NK, again, it's the number of cells carry K ECDNA copies. And then we look at the fraction of these cells, how many cells carry K copy numbers in the whole population. And then we calculate the Shannon diversity of these diversities. So if there's no diversity, then the Shannon diversity will be 0. And if there's a lot of cells equally carry different number of copies, then the Shannon diversity is higher. So if we look at our computational simulations, so the x-axis is the mean number of ECDN copies per cell. So it's a mean value for each cell population. And the y-axis is the Shannon diversity. We see, again, a fundamental difference between neutral selection and the positive selection. So if selection is neutral, you can see all the dots clustered in the low Shannon diversity. So here we only show n equal to 10 to the 5. But actually, if you have smaller values, they all clustered here. But if selection is positive, so here we show case S equal to 2, then we go to look at different population size. Again, because we are looking at the branching process, so population size here representing time as well. So you can see when time increases, then the Shannon diversity kind of shifted to high values as well. So which means that the distribution of the ECDN copies will shift to high values over time if selection is positive, but will cluster in a low Shannon diversity, or the distribution will remain in a low copy number distribution when selection is neutral. So again, we compared with the results from the cell line data. So how they have published this work in the glioblastoma. Well, they have the ECDNA distribution in this cell line, but the cell line population has different population sizes, so which means they can represent in different the times. So if you look at that, they have exactly one figure that is the x-axis, the mean number of ECDN copies per cell, and the y-axis is the Shannon diversity. And if we use our stochastic simulations to do ABC reference, and we found out when we have the simulation with parameter S equal to 3, will fit the data the best compared with the neutral selection. So we can't, of using this integration, think that for this cell line, for example, the ECDNA cells produce 3 times faster than the cells not carry ECDNA. So I think I'm coming to quite in the end of the talk. So again, we have some other work that's experimentally to confirming whether the ECDNA is positively selected or not, but is not so much integrated with the theory at this moment, so I'll just faster go through it. So Howard is a lab in Stanford. So what they have is that they have these CRISPR-R experiments where they can cut the ECDNAs. So they cut the cells in a random position in the genome. They can edit the chromosome DNAs, or they can cut the ECDNA structure, but not where the oncogene lies. Or in the last decade, for example, here they cut MUC, which this ECDNA carried the oncogene MUC, but they also added the MUC area. So they found out if they cut this ECDNA out, then the cell decreases. So which is kind of in an indirect way to show that these cells, when they carry the ECDNA, they have some fitness advantage. Once they lose the ECDNA, then they lose this advantage. They grow much less. So which is very similar here. If you look at the ECDNA, that the normalized copy number is that when they start experiments, and then they have look at 10 days later, how many copy numbers that the average population has compared with the initial population, then they see a similar pattern. So when they cut the ECDNA, then the copy numbers will first decrease and come back again after a while. So if you look at another experiment did by Paul's lab, so what they did is that they use treatment to treat target the cell has this ECDNA. For example, here is a glioblastoma with amplification on the EGFR gene, and then also see that the ECDNA copy numbers will shift it. So over time, the mean ECDNA copy numbers will decreases, which means that this kind of treatment introduce again a negative selection on the ECDNA copies, and they found out that if you take away the treatment, actually the ECDNA copies will be recovered again. So we have some continuous work in this direction to look at how can we include in the, for example, resistance mechanism of ECDNA and also the negative selection under treatment in our stochastic model. So I think up to here, I will end my talk. I would like to thank my collaborators that included the presentation, especially for all the people doing all these lab experiments, did a lot of work. And of course, with Yuri and Benjamin, who we three did the theory together. Thank you.
Thanks, Weini. Excellent talk, very concise, and which actually leaves some room for questions if people have some. But yes, I think it's been a great talk in terms of very simple, nice mathematical models, very well motivated in terms of the biology, nice integration of in vitro experimental data and mathematical model. So how much do you think that in vivo data could change this picture in your mind?
You mean in vivo?
Yes.
Yeah, so we actually have some patient data. So in our newest version, so in this version here, we had some data from Anton, from he's working. So they have some patient data, and where we can look at also referring the same fitness. And we actually also found out that the ECDNA is positive selected. Of course, we don't have the distribution data. So we only have the fraction of the cells and some other quantities that is not as detailed as the ECDNA copy distribution, but we all see a positive selection in the patient data as well.
Thanks. If anyone else has questions, I think Andre, maybe?
It's very fascinating work and very interesting phenomena. I'm sort of, I mean, there's so many kind of unknown and interesting questions still pending. One thing sort of from the data that you're showing, it's unclear whether one can really conclude that there's that observed distribution is just due to random partitioning of the circulating tumor DNA. And in particular, maybe you can address these questions. On one hand, it's unclear how this circulating tumor DNA amplifies. Is it like a chromosomal DNA, it's limited to a single round of replication, or are there multiple rounds of replication that these can undergo, like some viral DNA? And another important aspect is that a very kind of weird and interesting part of the behavior of this thing is that they can go in and out of chromosomes. And obviously, if one integrates into chromosome, that it kind of changes the whole dynamics of this random separation. So my question is like, how is there really how strong is the conclusion that the behavior of this extra-chromosomal DNA is entirely stochastic and not really influenced by ability to see, like, make many more copies than just like what the cell starts with, or by, you know, absorbing copies into chromosome and then passing them as sort of within a chromosome.
Very good question. Actually, we are writing a proposal together recently, and then one perspective we want to investigate is the reintegration of the ECDNA back to the chromosome. So they actually saw some, I think Anton said he saw some preliminary data that this might happening, and they want to investigate more of it. But in our computational simulations, for example, we didn't assume these complicated mechanisms. So we only assume random segregation. So when we only assume random segregation, the distribution we see kind of agree with the data cell line very well. So that's kind of bringing us confidence whether we need more complicated mechanism for it. But for some other experiments, for example, when they have the treatment data, and then the explanation need more complicated mechanisms. For example, how does the fitness depends on the copy numbers? But I think we kind of believe the random segregation is quite valid assumption here, because the cellular experiments they carried out in the staining. So when they have these daughter cells, they actually tested among quite different cell lines, patient-derived or just patient-derived cell lines or from different cancer types, but they all have this peak around 0.5. So we don't see one single cell line that don't have a random segregation hypothesis there. But the consequence is that then we kind of trusted from the simulations, because in our simulation, we didn't put any other assumptions, but they still agrees with the data relatively well. So
understand the simulations can recapitulate and could be a valid explanation for the observed. My question is, like, is it possible that one can get to a similar distribution through not through other mechanisms? And is there any way to kind of really come up with the non-trivial predictions from this random segregation in a way that can be tested experimentally? I think that would add like much more.
It's a good question. Then we should first model the other mechanisms to see whether the distribution will be similar or not. So
I think if you can come up with non-trivial predictions that are based on this random distribution, and you find that sort of your random distribution model accurately predicts these observations that you wouldn't be able to just things would be just simply selected or not, but the prediction of the something like involving subcloning experiments or something like that. I think it could be possible to test it, sort of to put more stress test on the model before having to consider more complicated scenarios, because there you have to rely on the bunch of assumptions without biological knowledge, hard to kind of specify and parameterize.
Exactly. I think, for example, currently, what you can see is that whether the process is completely random or not, obviously we can exclude that it's not completely random by the first experiments they did. But the other things is, what I think your question is whether you can have other mechanisms drive it. So then we need to kind of assume what kind of other things can bring fitness difference between cells carrying ECDNA or not. We didn't assume that the ECDNA copy doubling happening with independent of the cell division, for example, currently. So everything is happening together with the cell division, but we don't really have good evidence from their cell line data to say this happening in between the cells. For example, we don't see they say it's different as ctDNA, for example, because in the ECDNA that they actually come from parts of the chromosome DNA, so they kind of have this information that from chromosome instability. So if they see the daughter cells, they don't see that if you let the cell there without cell division, they see a huge change of the ECDNA copies. That's why we didn't do the assumption yet. But maybe our observation is limited. Maybe in the real cell, in the mouse or in the patient, maybe will be different. So we could think about in the future modeling, assume this happening, and then look at distribution whether it will make a huge difference or not. So this one I can't answer you because we didn't do it yet.
Cool. Audrey has a question. Audrey, you want to ask it yourself, or you want me to ask it for you? Because for the sake of time, Audrey, please go ahead.
Yeah, so I guess it's just sort of, it's probably very similar to what Andrew just asked, but I was thinking about how if this is a rare genetic event, you know, how common does it become in vivo and how quickly does that occur? Because that could perhaps tell you something about how stochastic the process truly is.
So I think actually the first paper that I cited in the talk is this Nature Genetics 2020 paper. They look at in the TCGA data set and they cross different cancer types. So it's quite common. So they found, for example, in the glioblastoma, 50% of the patient sample carries ECDNA. So I think it's not rare, but one question is not rare, but whether it's completely the driving force, for example, whether ECDNA alone can drive tumor is an unknown question yet. Because there are also another paper published recently talking about that cells actually has a mechanism to kill them, send a signal to kill themselves if they have some circular DNA in the cells. So if it's a totally normal cells, if they form this circular DNA, they will be killed. But so this might be that you have the cell need to have some mechanism to allow the ECDNA to be maintained, like to survive in the cell, and then drive the cell with cancer type. So we are thinking about, this is one thing that we want to look in the future, like whether ECDNA alone can drive the cancer dynamics or that you need the initial force. But our collaborators, they have some ongoing work shows that even in esophagus, for example, before they be developed to be tumor, in the pre-stage, they already have ECDNA there already. So this just make the question a bit early to say that ECDNA can arrive very early in the tumor development. But again, not answering the question whether it's the only driving force or it is a byproduct or second force to drive tumor evolution.
So that's really interesting. And I wonder if, does it seem like ECDNA is just more common in tissues that maybe have more genetic instability or a high turnover rate that could produce that?
So definitely that if you have more chromosome instability, is more likely to happen if it's a stochastic event as we. So, and also that you need to think about now people are starting work on it. Of course, everything they look at is oncogenes. But if it's chromosome instability, it can also happen that in non-oncogenes. So you might have other ECDNA elements in the cells that is not amplified oncogenes, so maybe not bring any fitness difference for the cells, also exist in the system. And then I guess I'm sure there will be new work. Once people finish the oncogene ECDNA work, they will look at whether they can use other tools to find other circular information. For theoreticians, will be super interesting. Then we can have examples in the cell lines that without fitness, and then look at whether the distribution will be very different compared with one with positive, because everything we test now is the data with the computation simulation, conforming to positive selection. But we would like to see that if you have no positive selection, what do you see as well? We had one experiment that just finished from the collaborator in Stanford. So they introduce randomly elements that's in the ECDNA, and without fitness, we do see the fraction of the cells not carry ECDNA decrease over time. So which means when S equal to 1, the fraction of cells carrying ECDNA become less and less. Yeah.
Okay. So yeah, thanks. We have a few more questions, and I'll be sort of asking people to ask those questions to you in the order in which I get. And I know Arturo has been raising his hand. I suppose that means he's got a question himself. Arturo?
Yeah, thank you. Hey, Weini, that was amazing. I really love the model. It was impressive to see everything just starting from first principles, which I love because you guide me through the whole process of this is what we're assuming, all the way to this is where we're doing the simulations. You mentioned that did I hear correctly? You're doing some agent-based modeling too?
Yes, so everything compared with data is from agent-based model because our theoretical prediction was under some limits. For example, our fraction the decay was under the limit of infinite population size. So we need to look at long-term dynamics. So we need the agent-based model to confirm some intermediate dynamics.
And I was wondering in the agent-based model, how many of these cells are you modeling?
So we model it up to 10 to the 11.
Oh wow, that's
modeling any spatial distribution. So, and also we don't carry any like mutation information. So it's just remembering how many copies you have. So for the simulation, it's relatively simple. Yeah.
So it's a non-spatial model. Do you think the space, putting in the space, could affect the results?
Yeah, definitely. I have a bit of work in spatial distribution of the chromosome DNA, and of course the ECDNA will be maybe even more like larger, because ECDNA dynamics happen faster. So the copy number, so the reason of this random segregation make it interesting is that if you have normal chromosome DNA, and to change the copy numbers is less slower than the ECDNA, because this random segregation can make the daughter cells suddenly become much higher numbers, and then this effect adds up in each cell division. So it makes the whole dynamics faster. So okay, also can see that if you have a spatial structure and you have a very faster dynamics, it will make a huge difference. It could be interesting to look at it because the people who work on the cell lines, they actually stay in the two-dimensional. Also, they have some kind of stomach cancer. As my limited understanding is kind of a two-dimensional cancer so if they have some data there, would be also interesting to have a look.
Yeah, I'd be really interested in seeing any results that come from that, even if they're like tentative results, because that could shed some light on the way that different cancers how the heterogeneity is distributed in different kinds of tumors.
Yeah, we are quite in the beginning. You see, we have this naive model, move to resistant. So next goal is go to the spatial.
And then once you have that, I think it'd be cool to model some sort of therapies and see how that affects the results. But yeah, it's great work. I'm very impressed. Thank you.
Okay, Asma, are you available to ask the question, or do you want me to ask it for you? Okay, so her question is, do treatments increase cytoplasmic DNA?
What do you mean? The treatment increase
cytoplasmic DNA.
So actually, I didn't talk too much about that because one of our collaborators from Berlin, for that treatment is that the ECDNA copy number increase. And the one that in Stanford, actually the ECDNA copy decrease. So it seems like the resistance mechanism has different. So some of them compensating to produce even more ECDNA copies, but some of them that the ECDNA just kind of get cut it if you get ECDNA, so they touch it in the cells carry high ECDNA, so they will get a negative fitness under treatment. So that's the last slide. What I show is that when we assume a negative fitness under treatment, and then the simulation can fit well again with the new distribution in the treatment very well. But we have another collaborator has the opposite result. So it's kind of believable that the different cell lines might have different mechanisms to overcome the
Very good. So we also have a question from Gosia. Gosia, I know that you can't talk, so you want to ask the question yourself?
Okay, I can do that. First of all, thanks for a really, really good talk. I'm just having a question about your results when it comes to fitting data and comparing it with your outcomes. So I noticed that you have one specific parameter, I think it was K3. Yeah. And I am wondering, because the nature of this whole process, it's, you are accumulating segregations, right? So I wonder if there's any threshold of some sort of specific tumor age after which your data is valid, because I cannot imagine this being valid for tumors in early development.
Yeah, so I think you asked a very good question. So when we refer in the ABC to say the selection is free, we want people to take not too serious about that. It has to be. So we actually, the major goal is to distinguish positive and neutral. So we are confident, because if it's neutral, the distribution won't shift. So it will be clustered with a lower diversity. The distribution will around with low copy numbers. And as soon as the selection is positive, over time, as you're right, it will dynamically increase. So which means time play a very important role. So if you have longer time, if that population run, the diversity will be higher. So in our simulation, we did need to try like time is a parameter, so population size is a parameter, and the selection intensity a parameter. So we found out in that distribution with k equal to 3 with a given population size fits the data very well. But as you said, we can't say this is exactly 3 because if you have different population size, maybe it's 5, maybe it's 2.5. But what we want to say is different with 1. So it's different with neutral. So it won't be fits the neutral fitness no matter which time is it.
Mm-hmm. Awesome. Thank you so much.
So Joel, you want to ask a question?
Sure, thanks so much. Weini, howdy.
Hi.
It is good to see you, and thanks so much for the presentation. My question is kind of, and it's just more sort of inviting you to, what would be sort of as one kind of looks through the crystal ball. So certainly convinced that having the extracellular DNA could provide additional coding, upregulation of metabolic pathways and whatnot by being able to make more, being able to translate more of the DNA. So you can certainly see how that would be advantageous. But now by the time you get to the experiments where people CRISPR out or manipulate the extracellular DNA, do you think by that point, the whole cancer cell has kind of co-adapted itself to this extracellular DNA? So in a sense, the advantage of extracellular DNA is also being coordinated with perhaps other co-adaptations. What do you think?
Well, I think because these cell lines, they're all independent experiments. So the CRISPR is just different experiments with the other cell lines we see. So that's why I think it's very nice that they have different cell lines to test. So not only the glioblastoma, well, in patients, most people see the ECDNA, but also in colon cancer, for example, that you only have 10% of patients carry the ECDNA, the dynamics still relatively persistent in different cell lines. And there was no real cell lines that we need to wait because, for example, in our simulations, we kind of, when we fit with the data, we need to pair different parameters because they are different cell lines. And the one thing we kind of assume that what different with the cell line is what is your initial condition. If you have cell lines, if you have patient data, you could say that, okay, you will arise from a rare event. So a single cell carry one single ECDNA copy and you simulate a long time to see how it fits the patients. But cell lines, quite often the experiments starting from maybe different initial conditions. You might have multiple cells carry different copy numbers. So your initial condition maybe is not already from your simulation. So to do this, actually one of our collaborators, he did this CRISPR, that's why he did this CRISPR experiments where he can introduce a single ECDNA copy in certain cells. So we will know the initial condition of what is the mean copy number for the cells carried ECDNA and what is the percentage. And then we can plug in that to simulate again and find out actually in our stochastic simulations, if you let the system run long enough, the equilibrium is very similar. So they kind of arrive into the fraction either far away from the neutral scenario case or not. And we don't want to make a very broad assumption to say this, as I said, this S value, the selection intensity is absolutely correct for this. So we currently just want to say we can distinguish positive and neutral selection by all this kind of dynamics because they are fundamentally different in the measurements. Yeah, but a very good question.
Oh, Weini, thank you. That's fantastic. What great experiments.
Yeah, Weini, thank you.
Credits. It's not we did the experiments.
Well, I know, but you're creating that lovely sort of lockstep between sort of the theory and the experimentation. That's lovely.
Yeah, we are lucky to meet the people. That's why the meeting seminar is so important. We're lucky to meet
the people.
It's
no lag. It's definitely lots of hard work and putting the effort where it is required. Weini, thank you very much. It's been a great talk and clearly it has led to a great conversation as well. It's very interesting research and it's really nicely integrated as well with experiments. It's something a lot of people here in IMO will be very interested to follow in the future as well. And thank you very much as well for making you sort of step in the middle of your maternity leave.
Thank you very much for having me here. This is really a great alternative. as I said, rocking the baby to sleep.
Yes. Well, thank you very much, and hope that some of us will be able to see you on stage maybe next year.
Yeah, hopefully to see you guys again in Tampa soon. I want to.
Oh yeah, in Tampa even better, especially in the next few months.
Yeah. Cool.
Bye.
Bye.